First of all, I would like to thank the organizers of this Congress for letting us organize this symposium. My talk will deviate a bit from the previous talk, and we're going to talk about longitudinal investigations of the cortex beyond thickness, area, and volume. As many of you may know, T1 weighted images, so structural MRI scans, have been used for quite a long time in studies using voxel-based morphometry methodology. And this technique allows for the quantification of gray matter density or gray matter volume at the voxel level. Now, and this kind of methodology has been used a lot in clinical studies, looking at case control designs, or looking at the relationship between these voxel-based measurements and, for example, symptom severity. Now, with the advent of surface-based morphometry, we were able to obtain more refined measurements of the cortex, such as cortical thickness and surface area. Now, the surface-based morphometry methods have also been used a lot in clinical designs, again, case control comparisons and so forth. But actually, the surface-based morphometry methods, they open up a way for new possibilities to look at other measurements that are maybe complementary to things like cortical thickness and surface area. And the aim of this talk is to give you some examples of surface-based morphometry-derived measurements and how they can be used in clinical and non-clinical settings. Okay. So, in this picture, you see the change of the cortex going from a lissencephalic aspect to a gyrencephalic aspect. And you can see the development of the sulci and the gyri. Now, new metrics that actually tap these huge neurodevelopmental changes that take place prenatally and early postnatally might be better tapping these early neurodevelopmental changes compared to like traditional metrics. So metrics that are specific to sulci and gyri might tap these changes more specifically than, for example, traditional measurements such as cortical thickness. So with this in mind, people started developing 3D measurements of cortical shape. And one of the most famous examples, as you may know, is this local gyrification index that was incorporated into the standard free surfer pipeline. And it gives you this local measurement of gyrified shape. And this has been used a lot in clinical studies, in clinical designs. And I'm putting just one example here on the bottom right. You can see a study that was actually discussed a few days ago by Dr. Drazan from the Institute of Psychiatry in London, where she explained that people with schizophrenia that did not respond to treatment had actually decreased gyrification compared to patients that did respond to treatment. And these kinds of studies indicate that there is some clinical significance to these measures of cortical shape. Now, the problem with this gyrification index that I just discussed is that it's a composite measure. So actually, if you want to know what drives these gyrification changes, you need to decompose this measurement. And there are numerous software packages to achieve this. And I'm just giving one example here. This is using the software package BrainVisa, which is freely available from their website. And on the top left image, you see a pile surface, and in blue you see what within BrainVisa terminology is called the sulcal median meshes. And these are like artificial meshes that follow the sulcal fundi. And these meshes can be used to extract sulcal-specific measurements. And you can actually parcellate, if you can see in the top row, the middle and the right figure, you can actually parcellate these meshes into regions that, as you would do normally for a free surfer parcellation. And you can also derive what is called a brain hull. The brain hull is like a net that envelopes the cortical surface without encroaching into its sulci. And this measurement gives you an indication or an estimation of exposed cortical surface. And this is interesting if compared to this non-exposed cortical surface, which can be derived from the so-called median meshes. On the bottom right, you see a schematic representation of the cortex with a number of measurements that can be derived. So this we did this in a previous study, and I'm just showing that these sulcal metrics that can be derived are things like sulcal width, sulcal depth, and sulcal length. There's also the thing of parcellation. We all know the standard free-surf parcellations. For example, the DAISY-CAN atlas, which is a default outcome if you run the free-surfer pipeline. But it might be that for your research question, these standard parcellations are not sufficient. So there are now software packages available and being developed as we speak that will allow you a more custom parcellation that might be more appropriate for your research design. And I've given one example here, which was a package by Liu in 2018 that allows for the segmentation of sulcal fundi and gyro crowns. As you can see in the bottom figure on the left-hand side, you get a parcellation, so a segmentation, a labeling of sulcal fundi in the bottom, and on the top, the gyro crowns. You can then combine these parcellations with standard free-surfer output and get a labeled version of these gyro crowns and sulcal fundi. And I will later explain a clinical example using these metrics. So now we are going to talk about a longitudinal study that we are doing. This is a study done in collaboration with the University Medical Center in Utrecht, where they obtained a fairly large longitudinal sample, a longitudinal single-site sample, consisting of healthy controls and patients with schizophrenia, where all subjects had 2 or more scans. And now we are analyzing this data in collaboration with the University Medical Center in Madrid. So an important point that I want to make is that these novel metrics, such as sulcal depth, width, and length, require the same kind of quality control as you would do for normal data as well, so traditional measurements like cortical thickness. Software packages like BrainVisa produce standard output, but if you are confident, if you want to be confident that these data are reliable, you will have to go back into the pipeline and check each step. You will need to do rigorous quality control at the end of the pipeline by visualizing all the output and manually editing if necessary. And even then, you will probably be in a position to discard a number of scans because they were just not sufficiently well processed. And this is exactly what we did for the longitudinal sample as well. So we actually discarded a number of images starting from 1800 and ending up with more or less 1100 scans. And an example of this this sulcal fundi and gyro crown parcellation can be given if we look at a publication from Waxdeel. And this publication included a schematic figure of the cortex. You can see this on the left-hand side, where in green you see the supragranular layers of the cortex, and in gray the infragranular layers. If we go from gyri to sulci, you can see the gray area decreasing. And what this means is that in sulci, these infragranular layers are underrepresented. And actually, the opposite is true for the supragranular layers, which are overrepresented in sulci with respect to gyri. Now, if schizophrenia is associated with supragranular thinning, which has been reported in post-mortem studies, then you might expect that this thinning is disproportionately large in regions of the cortex where these layers are overrepresented, which in this case would be the sulci. Now, our custom parcellations are an adequate way of assessing this hypothesis because they test the extreme regions in the gyri as well as the sulci. So we did that in the longitudinal sample, and what we saw more or less confirmed the hypothesis by Waagstiel. By using general additive mixed modeling, we show that in patients, which are here in blue, the sulcal fundi thickness is statically decreased in sulcal fundi, but the effect is not nearly as large in gyro crowns. We then went to a separate sample of first episode psychotic patients and found the same effect. So this means that there might be disproportionate cortical thinning that is more prevalent in sulci and gyri, which might be interpreted as indirect evidence for more prevalent thinning in supragranular layers in schizophrenia as compared to the more deeper layers, the infragranular layers. The second example is an example in a healthy sample. So we took only the healthy males and females from our longitudinal sample. And we wanted to ask the question, we all know that there's a cortical volume difference between males and females. What of our one-dimensional metrics, so I'm talking about sulcal depth, length, width, and cortical thickness, what of our one-dimensional metrics predicts best this difference between males and females in cortical volume? So we used a technique from normative age modeling, where we fit, again, we make a fit in females in this case. So in the figure you see on the x-axis age and on the y-axis cortical volume. And then in red you see the fit for females with age. And this is our normative model. So the females constitute our normative model. And we're going to see for every male the distance towards this fit. And as we also know, at each point of the normative fit, the standard deviations, we can actually calculate the distance and that standard deviations of each male towards the female fit. So the Z-score that we get represents the deviation of each male towards the normative fit. So we do this for cortical volume and our one-dimensional metrics to see deviation in which one-dimensional metric best predicts deviation in cortical volume. So if we put these, we check for collinearity, and then we put these deviations, predictors, in a linear mixed model predicting deviation in cortical volume. We see on the left-hand side in this effects plot that actually all but one of the one-dimensional metrics predict significantly this difference in the deviation in volume between males and females. But if we look at the explained variance per predictor, we can actually see that sulcal length by far, at least at the cortex level, predicts deviation in cortical volume, meaning that of our 4 one-dimensional metrics, it is really sulcal length that best represents the difference in volume between males and females. Now, the takeaway messages from this talk are that software packages such as Mindboggle, BrainVisa, and others can be combined with FreeSurfer to generate metrics that are not standardly given by the FreeSurfer output. Quality control for these other novel metrics is just as important as you would do for a traditional morphological study. And we think that these less traditional measurements can be complementary to these traditional metrics such as cortical thickness. Now, this work is a collaboration between Madrid and Utrecht, and I'm just the guy presenting the results, but there are actually loads of people involved, which I dearly thank. Thank you.